As you know the structure of DNA molecule, there are many electron-rich atoms present in the DNA, and these atoms are readily attacked by electron-seeking chemicals, and these electron-seeking chemicals are called electrophiles or electrophilic agents. Alkylating agents are a highly active group of electrophiles which transfer methyl, ethyl or alkyl groups to the electron-rich atoms in the DNA, and in this way they damage the DNA. Alkylation in DNA takes place mainly at 3 points, and these 3 points include the nitrogen and oxygen atoms external to the base ring system. You know there are nitrogenous bases, and there are some oxygen and nitrogen atoms which lie outside this nitrogen ring. So these atoms are the first site for such attack by electrophiles, and the second is the non-bridging oxygen atoms in the phosphate group. You know, phosphate group combines 2 nucleotides, and it has 4 oxygen groups. 2 of its oxygens are involved in the phosphodiester bridge formation, and the rest of 2 oxygen atoms are free. So the second site of such attack by electrophiles is these 2 oxygen atoms. And the third group is the nitrogen atom in the base ring system of nitrogenous bases, except that nitrogen which is attached with the deoxyribose. So you know deoxyribose is attached with the nitrogenous base through the nitrogen atom present in the nitrogenous base. So accepting that nitrogen atom, any other nitrogen atom is liable to attack by the electrophilic agents. So here you can see the different sites. So the first site may be this nitrogen, this oxygen, which lie outside the ring, outside the nitrogenous base ring. The second atoms or groups may be these oxygens which are not involved in phosphodiester linkage, like this and this. And the third one is the nitrogen which is present inside the nitrogenous ring system. It may be this nitrogen, this one, but it will not include this nitrogen or this nitrogen, because these 2 nitrogens, they are involved in the bond formation with the deoxyribose sugar, so they are not involved in this. Many different kinds of naturally occurring and synthetic chemical agents are known to transfer alkylating agents to DNA. So such chemicals, they cause alkylation damage to the DNA. Here are some examples of such chemicals, and they include dimethyl nitrosamine, dimethyl sulfate, which is called DMS, and NTG, N-nitro, N-nitrosoguanine, and MNU, N-methyl, N-nitrosourea, and MMS, dimethyl methane sulfonate. So these are some naturally occurring or synthetic compounds which cause alkylation damage to the DNA. So the product formed by attaching a chemical group to DNA is called an adduct. So sometimes these groups may be attached at different sites in the DNA, but in most of the cases they are involve only one site. So if the chemical group attaches to a single site on the DNA, then the product is termed as monoadduct.